Hello, my name is Bbumba Patrick, and I'm a biology teacher, and I'm here to take you through this topic of genetics, which is one of the topics which we cover at ordinary level biology here in Uganda. For more of these videos and so on, please don't always forget to subscribe to our YouTube channel, that is Christian Schools Owners Association, E-Class Teachers Uganda, and you'll be able to get me for any topic. Feel free to share as well as comment for any topic that you want me to work on in form of video. Now, this topic of genetics, we normally cover it in senior 3, and for this lesson 1 of genetics, I'm going to basically explain the genetic terminologies as well as looking at the monohybrid inheritance as according to the Mendelian genetics. Now, when I'm trying to introduce to you genetics, basically genetics is a branch of biology that deals with heredity and variation of organisms. And you can see here, we were having some good time with some students, and you could see that we are all totally different. Yes, we all have eyes, we all have a nose, we all have the lips, the teeth, the neck, hair, but much as we have the same body parts, but we are different. So you may ask yourself, what causes us to be different? What makes us to be different as human beings? Because we shall have short people, long people. What causes those variations? And you must know that such variations are normally passed on from one generation to another. And that is what we call heredity, whereby they're passed on from parents to the offsprings. And you can see here, this is a couple that had just wedded. And of course, after wedding, we know that what next is producing offsprings. And you can see these little angels here, these babies. So they must have characters which may resemble entirely one of the parents, or it may be a blend. So what causes that? Now, genetics tries to explain to you everything. In another term, we shall see that we have a term called chromosome. A chromosome, so chromosomes are normally many, and they're in every part of a human body. Because human bodies are built up of cells, like cells are the building blocks of the human body. Like every part of the human body you see is built of cells. Now, those cells, they have a nucleus inside. So I'm assuming this is the cell, which is the cell membrane. It will have a nucleus. Now, inside this nucleus, this is where we shall find the chromosomes. Now, normally the chromosomes appear as very thin thread-like structures inside, whereby you can't see them very well. But there is a process of mitosis. During mitosis, they condense and they become visible, and you can even count them. For each human being, in each of their cells, they have 46 chromosomes. Now, those chromosomes, they're the ones that make up, that hold the information to how the human being should look like. Some other term that you can think of is a gene, because normally we use that word gene. What is a gene? A gene is a basic unit of inheritance that determines the organism's characteristics. We have so many characteristics as human beings. Some are tall, some are short, some are brown, some are dark, so dark by the way. Some may have, let's say, 6 fingers or 5 fingers, or some. So what determines that? Okay, it's a gene. They are genes, and for every person, they have what we call a genome. A genome, that is the entire set of the genes of an organism. Now, for the humans, I think the entire set was mapped out by 2003. That's when the entire human genome was fully known. And also, there are the other organisms that we have, like rabbits, cows. They also have their own genome that determines how they look like, actually. Then another term that you want to look at is allele. Allele, plural alleles. It is an alternative form of the same gene. Normally, for every gene, there is an alternative form. For example, you could have a gene for tallness. Okay, I'm assuming if you can be able to see this chromosome here in this picture, and they're trying to section out a small portion of the entire chromosome. Remember, we have 46, and each of those 46 chromosomes determines certain characteristics of your body. And normally, those sections on each chromosome that determine the characteristics of an organism is what we call a gene. And you may find that there is one portion here on this one leg of the chromosome, which we call a chromatid, which is, let's say, a gene for tallness, and then another gene on the other leg, which is a gene for shortness. So that means for every gene, there is an alternative form. So much as there is an alternative form, they may be the same, of the same type, which we normally call homozygous. Of course, I will explain more about homozygous. Or they may be different, whereby one is represented by the capital T and then the other is represented by a small t. Then the locus is that specific fixed location on DNA where a gene or allele is located. We call it a locus. From location, then locus, that is that particular fixed location where the gene is located. And then there are some other words which I want us to understand. And these are all terms to do with genetics. Another term is homozygous. Homo means same. Homozygous. Now, zygosity or zygos has to do with the different alleles. So homozygous, it means same alleles, like they are the same alleles. So homozygous means having identical genes, one from each parent to form a particular character. And you can see here, I'm assuming we are using capital B to represent a certain character, which may be, let's say, black and black. So for this pair of chromosome, there is an allele here for black, which is capital B, and then another black, which is again capital B. But heterozygous means different. They are different. So having two different genes for a particular character, actually, you for better understanding, you may maybe call it allele. Yeah, let's call it allele. So having two different alleles for a particular character, and they're different. So you're having one allele, which is capital B, to represent the dominant character, and small b to represent the recessive character. Now, what do I mean by recessive and dominant? With the dominant comes from the word to dominate. It just simply means that allele that masks or suppresses the expression of an alternate allele. For example, if you're looking at these flamingos, you could have one purple flamingo mating with a violet flamingo, or if I am assuming it's red. So the baby that comes out, you are seeing that the baby is actually neither the blend, and the baby takes on the color of one parent, meaning that that the color that the baby has taken on from the parent is the dominant one. So that means this violet color will be the dominant allele. So because normally during fertilization, the male brings one set of chromosome and the female also brings one set of chromosome to form a diploid individual with two sets of chromosomes. Now that other allele which is not expressed, for example, for this purple flamingo, because it also brought its genes in forming this baby. So we call them a recessive allele. So a recessive allele is an allele that is masked by a dominant allele, meaning that it is not expressed outward in presence of a dominant gene. That is phenotypically. When I talk of phenotypically or phenotype, just means the physical appearance of an organism. How do you look? Okay, that is the phenotype. And the genotype just means the genetic makeup of an organism, just the genetic makeup. And in this case, you can see there's this animation which I got from somewhere. It's a very interesting animation. You hear this man with a unibrow. This man has a unibrow. That is, when you look at them, these are what we call eyebrows. These are eyebrows. But there are some people whose eyebrows connect, and such people, we say that they have a unibrow, just a single eyebrow, okay, because it connects. And you can see this man had a unibrow and the lady had a unibrow. Now, they did what they had to do so that they could have a baby. And guess what? When the man looked at the baby, first of all, the baby's color was different. It was looking like that one of the doctor, okay? Because the doctor is black and the man is white. Secondly, the guy said, wait, this can't be right. He wasn't seeing any unibrow. And he was like, Doc, where is his unibrow? Because he expected that he had a unibrow. And so the baby was supposed to have a unibrow. So how do you explain that? Genetics, okay? Another term that we shall have to look at is monohybrid cross. With a monohybrid cross, a genetic cross involving a single pair of genes, that is one character, for example, the character of unibrow, or tallness, or black color, or color. So, and parents may differ by just a single trait, whereby one parent may be black, that is when you're looking at the character or trait of color, and the other is brown, okay? And of course, in this picture, you are seeing this prominent basketballer who had a unibrow. I put this picture because some of you probably you've never seen someone with a unibrow. And an organism with similar copies of alleles for a given gene is said to have a homozygous genotype. Remember, genotype refers to the genetic makeup of an organism. So when you have the same alleles, then you are homozygous for that particular genotype. For example, capital T, normally we use letters to represent characters or traits. You can say maybe A could represent, let's say, albinism. That is, albinism simply means someone who lacks melanin in their skin. So they are normally called albinos, or R for red color of the eyes, and so on and so forth. While an organism with different copies of alleles for a given gene is said to be heterozygous, meaning that there are different alleles, whereby if one allele was represented with capital T, then the other is going to be represented with a small t. So this kind of genotype, we say that it is heterozygous for tallness. Then another term is having a pure breed. If you don't want to call it pure breed, you can call it true breeding, because it is basically that line which you call pure line or pure breeding. It's just homozygous. The parents are composed of the same alleles, purely the same alleles. So this is where the individual being crossed are homozygous. Those individuals, for example, in this case, I'm assuming we are using plants as the individuals. So you could have a white petaled plant and another white petaled plant. When you cross them, they are forming a white petaled plant. Okay, and then, but in this case, you could have case 2 whereby you cross a white petaled plant with another white petaled plant, and then you get a purple petaled plant. Then you can say, where is this coming from? Genetics. Then another term is crossing. With crossing, you don't mean that you cancel out. No. Crossing is like mating. We use the word crossing when we are meaning mating. Like, and we normally use this X, like you are seeing in this part here. And then another term is F1, which stands for first filial generation. Filio comes from the Latin word filius, which means the son. And so like the first sons, like the first children. So this refers to the set of offsprings obtained from crossing two pure breeding parents with contrasting characteristics. And then the other terms which I want us to look at is the term of selfing. I think people are normally used to selfie. When you talk of selfie, it means you get, let's say, a phone and then you use the front camera and then you take a picture of yourself. That is selfie. But in genetics, we mean that you cross offsprings from the same parent. I'm assuming, let me just use this example of mine, whereby this lady here is having two children, and if these children intermarry and they produce children, that is what we call selfing. But of course, in humans, that never works out because we call it incest, it's not allowed. So we use it, people use it in plants, we also use it in animals, especially those with good characters. Then also, we could also talk of test cross. A test is like you want to investigate something which you don't know. And here in this case, what don't you know? So this is a cross between an organism with an unknown genotype with a homozygous recessive organism. When I reach this part, I will explain to you why do we use a homozygous recessive organism so that we can determine the unknown genotype. Everything, I will explain this in the next video, which we shall be watching again on our YouTube channel. Then back cross, this is the mating of an offspring with one of its parents. You can imagine you have an offspring and then you mate it, let's say, with the parent. That is back crossing. So as to prove the genotype of the parents. Now, those are the most common terms as used in genetics. Now, I want to go and explain to you the Mendelian genetics. But before we can, and which include the monohybrid inheritance. So before we can go to the monohybrid inheritance, I want us to first talk about this guy who is the pioneer of genetics. His name is Gregor Johann Mendel. Gregor Johann Mendel is the pioneer of genetics. What do we know about this guy? He was an Australian monk born in what is now called the Czech Republic, 1822, and is the son of a peasant farmer. He was the son of a peasant farmer. He studied theology and was ordained a priest of the order of St. Augustine. And you can see he's putting on, he's dressed like how priests normally dress. So he did go to university, the University of Vienna, where he studied a lot of botany and he studied scientific methods. He worked with pure lines of peas for 8 years. You can imagine 8 solid years working on peas. For some of you who don't know peas, there is a certain type of, we normally eat them here in Uganda. Locally, it's called kao. And pure lines means that you are looking at homozygous individuals. Now, prior to Mendel, hereditary was regarded as a blending process, and offsprings were essentially a dilution of the different parental characteristics. So people didn't know so much about genetics until this guy came and gave us his work. Now, what did this guy study in peas? Now, this guy, he studied 7 characteristics of peas. He looked at smoothness of the seed, whereby, shape of the seed, where some seeds were smooth, others were wrinkled. He looked at seed color, where some seeds were yellow and some were green. He looked at the pod shape, inflated and constricted. He looked at the pod color. Some pods of peas were green and some other pods were yellow. He also considered the color of the flower, whereby some had purple flowers and some had white flowers. He also looked at the flower location, where in some plants it will be located at the leaf junction, whereas in others at the tip. And then also plant size, whereby some would be so tall peas, and others would be short, which he called dwarfs. You can see, as a scientist, he was able to look at all these characters. Imagine, as a scientist, you're supposed to be so observant and so that you are able to look at each detail. That's what science is all about. So he crossed these plants for his experiments, and he did a lot of mathematical work. Actually, he was the first scientist to explain biology using mathematics. So what we are going to do next is actually using mathematical models to explain genetics. And normally we ask, why do you think Mendel used garden peas, cow? Why did he? These are the peas that I'm talking about. One, they occurred in many varieties. There were so many different varieties with distinct characters: tall, short, green pod, yellow pod, and so on and so forth. Also, they were easy to cultivate, and all offsprings were fertile, because it is important, because you need to keep on producing and producing and producing. They also had a short life cycle, so they reproduce quickly. You want a plant within 3 months, it is giving you results. Within 3 months, it is giving you results. But imagine if it were taking 2 years, you wouldn't have done this good job. Also, the plants also had many contrasting characters, as I've already talked about it. Now, going forward, how did Mendel do this? How did he? Now, he crossed, if you can look here in the upper right corner, he crossed pure tall peas, pea plants, with pure short pea plants. Remember, pure means homozygous. You're dealing with homozygous individuals, and all offsprings were tall in the F1, that is first video generation. Those were the first offsprings. So meaning that if you have crossed pure tall and pure short, and then all over, all what you are getting is tall, it means that the tall character is dominant character because it masks the short character. So that means the short character is the recessive character, because the recessive character is not expressed phenotypically in the presence of a dominant gene. Remember, phenotype means outward appearance. So you don't see the peas as short, no. You only see tallness, meaning that tallness is dominant because it masks the other recessive, that is the short character. And normally for the dominant character, we represent using a capital letter, while the recessive character we represent using small letters. That is standard. And when you're doing it, you first say let capital T. You just think of any letter which suits your character. And in this case, we are choosing capital T to represent the allele for for tallness, and small t to represent the allele for shortness. And these are just structures. So when you're doing, first of all, you have to first write the parental phenotype. How do the parents look like? In examination, you always have to begin with the parental phenotype, meaning how do they look like? So you cross the tall pea plant with a short pea plant. Then afterwards, because what you're seeing outward is not what determines the character. Actually, it is what is hidden inside, that is the genetic makeup. So next you go to the parental genotype, those that you are crossing. So you're crossing pure lines, that is capital T, because they're homozygous for tall, and then small t, homozygous for short. And so from getting the genotypes, when these are mature, they produce gametes. So, and under the process called meiosis, so you write meiosis. This meiosis will produce for us the gametes, which are normally, according to Mendel, like in each gamete, it can accommodate only a single set of allele. So here you have a single gamete goes in, a single character goes into a single gamete. So the gametes all have single, and that's why we are putting this N, meaning haploid. So that one N means the gamete is haploid, meaning that it is having one set of chromosome from the parent, haploid. And we are putting 2N here to represent diploid, meaning that these individuals, they have 2 sets of chromosomes, and normally one they got from their dad and another from their mom. So when you're having these gametes, they must undergo fertilization, where gametes on one side, each gamete here, either gamete may fuse with the either one on the other side. That is according to the second law of Mendelian genetics. So you're having random fertilization, like It's just random, where capital T will fuse with a small t to form capital T, small t. Now, this individual is heterozygous. You can also have this gamete fusing with another gamete here. Again, this individual is going to be heterozygous. Again, you can have this capital T fusing with this small t. Again, the individual is heterozygous for tall. Again, you'll have this capital T fusing with another small t. The individual will be homozygous for tall. Sorry, heterozygous for tall. Okay, and so those are the ones that you get. So that's how the F1, the first offspring will be like genotypically. But then this is not what you see. What you see is their phenotype. You're going to see them when they're all tall. So these were the first results of Mendel. But then he further crossed these F1 offsprings. Now, when you cross the F1 offsprings, that is, you'll have the parental phenotype, that is F1 phenotype in this case, because you are looking at these offsprings which we got earlier. So we shall have the F1 of phenotype giving you tall P with tall P. And never forget to put this X because it means they are mating. So you are mating tall P's with tall P's. And then how does the genotype look like? The F1 genotype will have a heterozygous tall. That is one, the allele are not the same, they are not similar. So you have a capital and a small allele, capital and small allele. And you're crossing them, that means they are mating. Now, these individuals, the genotype, they must produce gametes under a process of meiosis. So meiosis will occur, and then that gamete will be formed from one of the parents, and another gamete will be formed from that parent. Again, under meiosis, I'm assuming this parent was the dad, so these are sperms. Maybe I could put a small tail to show they are sperms. So these are sperms, and also this individual will produce that egg, and that individual will produce this egg. So these sperms must undergo fertilization to fuse with these eggs from the female parent. So those gametes, you have random fertilization, whereby this sperm fertilizes this egg to form this individual with capital T, that is purely tall. And then that sperm will fuse with that egg to form that individual who is heterozygous. I think you already seen how variations come to be among organisms. Then that sperm will fuse with that egg to form that heterozygous individual. Then again, that egg will fuse with that, that sperm will fuse with that egg to form that short individual. So this genotype you can't see, but you will need the phenotype to show you that this is going to be tall, this is going to be tall, this is going to be tall. So you're having the ratio of 3 tall plants to 1 short. So this one is going to be short because the gene for shortness is only expressed in absence of a dominant gene. And that's why even when here the gene for shortness appeared, we did not call this plant tall because it is masked by this capital T. So you can't see it, okay? So this is the monohybrid ratio as put forth by Mendel. So the ratio of the dominant recessive phenotype is 3:1, and we call this the monohybrid inheritance. Now, there are so many examples, and Mendel, when self the plants of the F1 generation and obtained an F2, so this is the F2 generation, he got this ratio of 3:1, which we call the monohybrid ratio. Now, there are so many other characters which are exhibited in this format, and one of them is albinism. I don't know whether you've ever seen an albino. With albinism, we mean a person who lacks melanin in their skin, okay? So this condition, because a person is not able to produce melanin, so they will appear white, the hair will be white, they will not have pigment in their iris, they will all be, and by the way, both whites, that is the bazungu, and blacks, the Africans, we can all be produce albinos. Okay, so albinos will have light hair, white hair, pink eyes. They are very sensitive to bright light, and that's why sometimes they have to use sunscreens, whereby they must screen like that sunscreen Vaseline is the one which acts as the melanin. It's the one that acts as the melanin. So we shall be able to understand more of them slowly. Now, this character is recessive, and that's why you don't normally see it in the population, meaning that for you to be an albino, it means you must have double character. You must have double character for albinism. So here we say that let A be the allele for normal skin color, and let small a be allele for no skin color. And normally the genotype, when you get a genotype of capital A, meaning that you being able to produce melanin, it is what we call its dominant, capital A. So you will have a normal skin color. But when you are heterozygous, like capital A, small a, you will be just a carrier. And by the way, any of us can be a carrier for albinism, but you don't know unless you meet someone who is a carrier, and that's when you can produce an albino. When you have small a, it means that you're going to be an albino. So for you, you are homozygous. That's your genes are. And we can further work out a problem here, whereby the problem is what would be the offsprings if an albino marries a normal person? What would be the offspring? Now you say let capital A be the allele for normal skin and small a be the allele for no skin color. Remember, we always have to first write the parental phenotype. So the parental phenotype, or you can call them F1 phenotype, we shall have a normal skin with an albino. So you're having what would be, actually here, even if you don't put F1, even if you just say parental phenotype, because that's the starting, this is the starting parental phenotype. So you are having an albino marrying a normal person. So a normal will have capital A, that is the genotype, and an albino will have small a. So when you cross them, remember these genotypes must undergo meiosis to produce the gametes, which will further undergo fertilization. So this normal person will produce that sperm, and then he will also produce another sperm, and then the other albino will produce the egg. I'm assuming the wife is the albino, and also another egg. Now those eggs will undergo, those are the gametes, sperm and egg. So they will undergo random fertilization, where you have a normal but carrier genotype, another carrier genotype, another carrier genotype, another carrier genotype. And you can see capital A is in every genotype. Remember, capital A is the dominant and is the one for normal skin, meaning that all the children that will be produced from a couple of normal skin and an albino, they will all have normal skin. They will not produce any albino, okay? And but if you are having scenario 2, what would be the offsprings when two individuals who are married, who are carriers of albinism, get married? So you could have a black, like normal skin color person, wife, and normal skin color husband, but they are carriers. Again, you say let capital A represent allele for normal skin color, let small a represent allele for no skin color. Remember, we begin with parental phenotype. So this should be parental phenotype, normal skin with normal skin. Now there, you've not gone for genetic counseling, you don't know, you get your wife, and now finally you reaches a time you produce sperms. So this you produce these sperms, you produce this other sperm, then your wife produces those two eggs, and then in the process of fertilization, you produce an individual. The first baby, the baby is normal, normal skin. You even carry out a baby shower and all those things. The second baby, the baby is normal. You celebrate. The third baby, the third baby is normal. You celebrate. Then the fourth born comes out as an albino. That's when you say, kumbe we were carriers. So here you'll have a ratio of one normal person and two carrier individuals and one albino, which phenotypically will come out as a 3:1 ratio still, according to the Mendelian monohybrid inheritance, that when you cross the F1 offsprings, okay, when you cross the F1 offsprings, you'll always get a 3:1 ratio. Okay, and then there are some other characters still, and another character which is exhibited by monohybrid inheritance is sickle cell anemia. That condition where a person's blood, body red blood cells are sickle-shaped, and so they are not able to transport oxygen in his body well. Also albinism, which we have just talked about. There's also achondroplasia. Achondroplasia is this condition that you are seeing here. You would have these characters. Another one is cystic fibrosis, whereby this one normally comes on in an advanced stage, like your liver produces, not liver, sorry, our lungs may produce certain fluids, and so you may get some infection and you also die. So those are some of the characters, and feel free to undergo and take on this quiz, which I'll also share in Google Classroom, and then you'll also be able to try it out, and I'll mark it. And I hope that's all. And please don't forget to always subscribe to our YouTube channel, which is Christian Schools Association of Uganda, A-Class Teachers Uganda, so that you can have more of these classes. And that's it. Have a lively day.